Good afternoon, dear students. We are glad to see you on the course of biology. Today we have the third practical lesson. The topic is structural functional organization of the eukaryotic cell nucleus. The purpose of this lesson is learning structural and functional organization of the eukaryotic cell nucleus. Key questions which will be discussed during this lesson are presented on this slide. The cell is an elementary self-regulating structural and functional unit of tissues and organs. It contains the process underlying the energy and constructive supply of structures and the level of functioning of the body. The cell nucleus consists of a nuclear envelope which separates it from the cytoplasm, chromatin, and nucleus or nucleus, and other products of synthetic activity. Nuclear protein backbone, the matrix, and karyotype, also called nuclear sac. The main component of nuclear chromatin is a structure that performs genetic function of a cell. Almost all genetic information is covered in chromatin DNA. The nuclear envelope performs a complex barrier receptor function as well as transport and scaffold. The non-chromatin nuclear protein backbone, or matrix, provides not only the special position of chromosomes but also participates in functional activity. One of the chromosomal regions that determine the synthesis of RNA and the formation of cellular ribosomes is the nucleus or nucleus. In addition, various ribonucleoprotein structures containing different types of RNA are found in the nucleus in connection with chromatin and matrix. Between all these components, there is a liquid phase of the cell nucleus, karyoplasm, in which many processes which are associated with both nuclear metabolism and a nuclear transport of proteins and RNA occur. Nuclear pores or nuclear pore complexes are large protein complexes that penetrate the nuclear membrane and transport macromolecules between the cytoplasm and the cell nucleus. The transfer of molecules from the nucleus to the cytoplasm and in the opposite direction is called nuclear cytoplasmic transfer. According to electron microscopy data, the nuclear pores cross-sectionally have the shape of an eight-spoke carved wheel. That is, they have an axis of symmetry of the eighth order. These data confirm the fact that nuclear purine molecules are present in the nuclear pore in a multiple of 8. The channel that is permeable to molecules is located in the center of the structure. Nuclear pore complexes are enfolded in the nuclear envelope with the help of a transmembrane part from which structures called spokes are turned to the channel lumen by analogy with the spokes of a cart wheel. This whole part of the pore, built of 8 domains, is bounded from the cytoplasmic and nuclear sides respectively by the cytoplasmic and nuclear rings. Attached to the nuclear ring are protein strands directed into the nucleus or nuclear filaments, to the ends of which a terminal ring is attached. This whole structure is called the nuclear basket. Attached to the cytoplasmic ring are strands directed to the cytoplasmic filaments. In the center of the nuclear pore, one can see an electron-dense particle, a slip or a block. The nucleus is a non-membrane and a nuclear subcompartment inherent to all eukaryotic organisms without exception. It is a complex of proteins and ribonucleoproteins that form surrounding regions that contain RNA genes, nucleolar organisms. The main function of the nucleus is the formation of ribosomal subunits. In the nucleus, 3 main structural components are distinguished, responding to different stages of ribosome biogenesis. There are fibrillar center, dense fibrillar component, and granular component. At the beginning of mitosis, the nuclei are disassembled, and at the end of mitosis, they are assembled again. Current There are data on the participation of nuclearly encoded not related to ribosome biogenetics. For example, in stress response, assembly of signal recognition protocols. In addition, the nucleolus interacts with many viruses. The nucleolus is involved in the development of many human diseases, including cancer and possibly neurodegenerative and autoimmune. Chromatin, from ancient Greek color, stains, is a nuclear protein that forms the basis of chromosomes. Consists of DNA and proteins, mainly histones. Chromatin is found inside the nucleus of eukaryotic cells and is part of the nucleoid nucleus. It is in the composition of chromatin that genetic information is realized, as well as DNA replication and repair. Unlike prokaryotic cells, the DNA containing material of eukaryotic chromatin can be in two alternative states: decondensed in the interphase and in the non-dense state during mitosis as part of mitotic chromosomes. In non-dividing or interphase cells, chromatin detected under a molecular microscope can easily fill the volume of the nucleus or be located in a separate blocks that calls chromocenters. Often it is especially clearly detected on the periphery of the nucleus, parietal, marginal, minimum-grain chromatin. All forms interleaving of rather thick, about 0.3 nanometer, and long strands inside the nucleus, forming a semblance of an intranuclear network. Such nuclei are found in plant cells, for example. Chromatin of interphase nuclei is the new living bodies, chromosomes, which at this time lose their compact form, loosened, and decondensate. When it is completely decondensed, these forms are called diffuse chromatin. With the complete loosening of chromosomes in the interphase nucleus, areas of condensed chromatin, heterochromatin are visible. Heterochromatin, active chromatin, is chromatin region that retain the despolarized state of elementary deoxyribonucleoprotein filaments in the western nucleus, for example, in the interphase, like other regions that retain the helical state, or heterochromatin. Heterochromatin also differs from heterochromatin in its ability to intensively synthesize ribonucleic acid, or RNA, and a higher content of non-histone proteins. In addition to deoxyribonucleoprotein filaments, it contains ribonucleoprotein particles, which serve to complete the maturation of RNA and transfer it to the cytoplasm. Chromatin contains most of the body's structural genes. Histones are a vast class of nucleoproteins that perform two main functions. They are involved in the packaging of DNA strands in the nucleus and in the epigenetic regulation of nuclear processes such as transcription, replication, and repair. There are 5 different types of histones: H1, H5, H2A, H2B, H3, H4. Histones H2A, H2B, H3, H4, called core histones, form a nucleosome, which is a protein globule around which a DNA strand is wound. Histone H1H5, called linker histone, binds to the outside of the nucleosome, fixing a DNA stranding. In chromatin, histones account for about 25–40% of the one weight. Due to the high content of lysine and alanine, histones exhibit strong basic properties. Histones are in direct contact with DNA and are able to neutralize the negative charge of phosphate groups in DNA due to the positive charge of amino acid residues. The amino acid sequence in these proteins is conservative and therefore doesn't differ in organisms of different taxa. or histones. Histones are present in the nuclei of eukaryotic cells. Bacteria do not have histones, but they are found in outer of the eukaryotic group. Histones were discovered in 1884 by the German biochemist Albert Kossel. Two molecules of each of the histones H2A, H2B, H3, and H4 make up an octamer emblazoned with their 146 base pair DNA segment that forms 1.8 helix turns over the protein structure. This particle with a diameter of 7 nanometers is called a nucleosome. The DNA region connecting nucleosomes are not directly in contact with the histone octamer interacts with the linker histone H1. The length of the DNA fragment per nucleosome varies and averages 200 base pairs. In this case, 146 base pairs are directly connected to the nucleosome and the remaining several times connect to neighboring nucleosomes. DNA and nucleosomal histones are firmly connected. In each nucleosome, 142 hydrogen bonds are formed between DNA and the histones that make up it. Almost half of these bonds arise between the main chain of histones and the phosphodiester groups of the sugar phosphate backbone of DNA. In addition to hydrogen bonds between DNA and proteins, nucleosomes hold together numerous hydrophobic interactions and cell bridges. For example, the positive charges of the amino acids lysine and adenine, which are enriched in histones, can effectively neutralize the negative charge of the DNA backbone. These numerous interactions probably explain why DNA of the virtually milliseconds can be associated with a nucleosomal octamer. Core histones H2A, H2B, H3, and H4 are small proteins with molecular weights of about 10–15 kilodaltons, the composition of which is extremely enriched in the positively charged amino acids lysine and adenine. Positively charged amino acids are concentrated mainly in the amine and carboxyl terminal portions of the core histone molecules called tails. Histone tails, about 15–30 amino acid residues long, are not organized into any pronounced secondary structures. Histone tails, primarily the end tail, play a key role in the epigenetic mechanisms in which these proteins are involved. In the central, most conserved regions of the polypeptide chain of core histones, residues of hydrophobic amino acids prevail. It is these central regions that are involved in the formation of the nucleosomal octamer onto which DNA is bound. The central region of all nucleosomal histones has a characteristic secondary structure with an extended alpha helical domain, which is flanked on both sides by domains containing one loop and one short alpha helix. This special structure is called the histone fold domain. Thus, nucleosomal histones contain a central structure, three-stranded histone fold domain, and unstructured amine and carboxyl tails. Histones H3 and H4, and also H2A and H2B, recognize each other pairwise. Spiral domains interact with each other, forming structures called handshake, resulting in the formation of heterodimers H3, H4, and H2A, H2B. From the third dimer, in turn, the tetramer H3, H4 is formed. The tetramer H3, H4, and two dimers H2A, H2B make up the histone octamer, the core of the nucleosome. The nucleosome is wedge-shaped. Its narrow part is H3H4, 2 parts, and the wide part consists of 2 dimers, H2A, H2B, which are located on the sides of the tetramer H3, H4, 2 parts, and do not interact with each other. Of all the DNA which is wound around the nucleus tetramer, about 80 base pairs are linked to the tetramer, and about 40 base pairs are linked to the H2A, H2B dimers, tetramer H3, H4. Each type of histone, except for histone H4, is a group consisting of canonical histones and histone variants. The role of histone variants is to preserve the nucleosome pattern of chromatin, increase or decrease its resistance, create a special context in each specific chromatin region, and thereby control the process of transcription, replication, and repair. The total length of DNA in human cells is about 2 meters. The diameter of the cell nucleus is about 7 microns. Considering that each chromosome is represented by a separate DNA molecule, the level of DNA compaction is more than 6,000 times. Different levels of DNA packaging are distinguished. Different sections of DNA can be packaged to varying degrees. The first level of DNA compaction is nucleosomal. If chromatin is exposed to nucleases, then DNA undergoes decay into regularly repeating structures. These repeating structures are H molecules of histone proteins, or octamers, and DNA wrapped around this octamer. Such a complex nuclear protein particle is called the nucleosome. In it, histones form a protein core on the surface of which there are almost 2 tons of DNA. Between the nucleosomes there remains a section of DNA not associated with histones, which connecting to adjacent nucleosomes passes into the DNA of the next nucleosome. This level of compaction is sometimes referred to as bits on a string, where the bits are the nucleosomes and the string is the DNA. The diameter of the nucleosome is about 11 nanometers. The first nucleosomal level of chromatin compaction plays a regulatory and structural role, dividing the density of DNA packing by 6 or 7 times. The second level of compaction is a 30 nanometers fibril. In mitotic chromosomes and in interphase nuclear chromatin fibrils with a diameter of 25-30 nanometers are detected. A solenoid type of nucleosome packing is distinguished. A thread of tightly packed nucleosomes, 10 nanometers in diameter, forms turns with a helix each of about 10 nanometers. About 6 or 7 nucleosomes per turn of such a supercore. As a result of such packing, a fibrillum of a spiral type with a central cavity appears. Chromatin in the nucleus has 25 nanometers fibrils, which consist of contiguous globus on the same size, but called nucleomers. These nucleomers are called superbeads. The main chromatin fibril with a diameter of 25 nanometers is a linear alternation of nucleomers along the compacted DNA molecule. As part of the nucleomer, 2 turns of a nucleosomal fibril are formed, 4 nucleosomes in each. The nucleomeric level of chromatin packing provides 40-fold DNA compaction. Nucleosome and nucleomeric or superbead levels of chromatin DNA compaction are carried out due to histone proteins. Next level of compaction is loop domains of DNA, the third level of structural organization of chromatin, chromomeric. In the higher levels of chromatin organization, specific proteins bind to special regions of DNA, which forms large loops or domains at the binding sites. In some cases, there are plots of condensed chromatin, rosette-like formations consisting of many loops of 50 nm fibers connecting in a dense center. The average size of the circuits reaches 100 or 150 nanometers. Rosettes of chromatin fibers cause chromomers. Each chromomer consists of several nucleus-containing loops that are linked at a single center. Chromomers are linked to each other by regions of nucleosomal chromatin. Such a loop domain structure of chromatin provides structural compaction of chromatin and organizes functional units of chromosomes, replicons, and transcribed genes. And the next one is denser pattern of DNA, chromatid at chromosome level, is achieved through further compaction of chromomers and is observed in dividing cells. In them, the chromosomes are so dense that they become visible under a light microscope as separate formations. In a non-dividing cell, chromosomes are despolarized or decondensed. The boundaries between them are not visible, and their diffuse material is called chromatin. An electron micrograph in a plant cell shows darker and denser areas of the heterochromatin and lighter, loose areas of aerochromatin. Aerochromatin is transcriptionally active. Heterochromatin is mainly silent regions of DNA, and telomers and centromeres are structural elements of chromosomes that do not contain genes. Centromeric and telomeric regions belong to obligatory heterochromatin. Optional heterochromatin may or may not be formed in a given region of DNA in a particular cell type. Chromosome structure. The eukaryotic chromosome is formed from a single and extremely long DNA molecule that contains a linear group of many genes. The necessary functional elements of the eukaryotic chromosome are the centromere, telomers, and points of replication initiation. The points of the beginning of replication, or replicon origins, and telomers located at the ends of chromosomes allow the DNA molecule to replicate efficiently, while in centromeres, sister DNA molecules attach to the mitotic spindle of division, which ensures the exact divergence across daughter cells in mitosis. In the course of the cell cycle, the appearance of the chromosome changes. In the interphase, these are very delicate structures that occupy separate chromosomal territories in the nucleus, but are not noticeable as separate formations during visual observation. In mitosis, chromosomes are transformed into densely packed elements capable of resisting external influences, maintaining their integrity and shape. Take the chromosomes at the prophase, metaphase, or anaphase stage of mitosis that are available for observation with a large microscope. Mitotic chromosomes can be seen in any organism whose cells are able to divide by mitosis, with the exception of some organisms, for example, Staphylococcus cereus, whose chromosomes are too small. Typically, mitotic chromosomes are serial microns in size. For example, the largest human chromosome, chromosome 1, has a length of about 7 or 8 nanometers in metaphase and 10 nanometers in prophase of mitosis. At the metaphase stage of mitosis, chromosomes consist of two longitudinal copies called sister chromatids, and which are formed during replication. In metaphase, chromosomes, sister chromatids are joined at the primary construction called the centromere. The centromere is responsible for the divergence of sister chromatids into daughter cells during division. The centromere assembles the electrophore, a complex of um, a complex or protein structure that determines the attachment of the chromosome to the microtubules in the fission spindle, the nucleus of the chromosome in mitosis. The centromere divides the chromosomes into two parts called the arms. In most species, the short arm of the chromosome is denoted by the letter P, and the long arm by the letter Q. Chromosome length and centromere position are the main morphological features of metaphase chromosomes. Depending on the location of the centromere, three types of chromosome structure are distinguished: acrocentric chromosomes, in which the centromere is located almost at the end, and the second arm is so small that it may not be visible on cytological preparations; submetacentric chromosomes with arms of unequal length; and metacentric chromosomes, in which the centromere is located in the middle or almost in the middle. And another type, telocentric, is very rare. Here, chromosomes have only two shoulders, in general, and one shoulder at each part of chromosome. So not two shoulders, like for example here, but only one here and here. This classification of chromosomes based on the ratio of shoulder length was proposed in 1912 by the Russian botanist and cytologist Navashin. In addition to the above three types, Navashin also distinguished telocentric chromosomes, that is, chromosomes with only one arm. However, according to modern concepts, there are no true telocentric chromosomes. So, as we said before, the second shoulder, even if it's very short and invisible in the ordinary microscope, is always present. An additional morphological feature of some chromosomes is the so-called secondary constriction, which outwardly differs from the primary in the absence of a noticeable angle between chromosome segments. Secondary constrictions come in different lengths and can be located at different points along the length of the chromosome. Secondary constrictions usually contain nucleolar organizers containing multiple repeats of genes encoding ribosomal elements. In humans, secondary constrictions containing ribosomal genes are located in the short arms of acrocentric chromosomes. They separate small chromosomal segments called separates from the main body of the chromosome. Chromosomes which have a separate are usually called set chromosomes, so they are without DNA. The basis of the chromosome is a linear DNA macromolecule of considerable length. In the DNA molecules of human chromosomes, there are from 50 to 235 million pairs of nitrogenous bases. The total length of DNA from one human cell is about 2 meters. At the same time, a typical human cell nucleus, which can be seen only with a microscope, occupies a volume of about 110 nanometers in the third, and the human mitotic chromosome doesn't exceed 5 or 6 nanometers on average. Such a compaction of genetic material is possible due to the presence in eukaryotes of a highly organized system of packing of DNA molecules both in the interphase nucleus and in the mitotic chromosome. It should be noted that in eukaryotes, in proliferating cells, there is a constant regular change in the degree of chromosome compaction. Before mitosis, chromosomal DNA is compacted 105 times in comparison with the linear length of DNA which is necessary for the successful segregation of chromosomes into daughter cells, while in the interphase nucleus for successful transcription and replication, the chromosome must be decompacted. At the same time, the DNA in the nucleus is never completely eliminated and is always packed to one degree or another. Thus, the populated decrease in size between the chromosome in interphase and the chromosome in mitosis is only about 2 times in yeast and from 4 to 50 times in humans. DNA packing into chromatin provides multiple contraction of the linear dimensions of DNA, which is necessary for its placement in the nucleus. It takes place in several stages. The most studied are the first 3 levels of packing. First of all, DNA binding onto nucleosomes with the formation of the nucleosome strand, 10 nanometers in diameter, compaction of the nucleosomal strand with the formation of the so-called 30 nanometers fibril, and the third is folding of the later into giant loops from 50 to 200,000 base pairs on the protein skeletal structure of the nucleus, the nuclear matrix. Some researchers believe that one of the most recent levels of packing into the mitotic chromosome is the level of the so-called chromatin, the thickness of which is about 0.1 and 0.3 nanometers. As a result of further compaction, the chromatin diameter reached 700 nanometers by the time of metaphase. The significant thickness of the chromosome diameter 1,400 nanometers at the metaphase stage allows finally to see it through a light microscope. The condensed chromosome is by the letter X, often with unequal shoulders, since the 2 chromatids resulting from replication are connected to each other in the central region. For more information on the fate of chromosomes during cell division, you can see the articles about mitosis and meiosis to understand this process better. In 1960, in the American city of Denver, the first international system of cytogenetic nomenclature of human chromosomes was created, which ensured the international standardization of chromosome studies at the initial stages of the formation of human cytogenetics. The chromosomal set of karyotype or karyotype of a person includes 46 chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes, XX in females or XY in males. The Denver classification of chromosomes was based on their morphological characteristics, size, shape, and position of the primary transcription centromere. According to this nomenclature, chromosomes are numbered from 1 to 23 in decreasing order of their length. From 1st to 22nd are autosomes, and 23rd are parsexual chromosomes. The largest human chromosomes, which have the first numbers, are on average 5 times longer than the smallest 21st and 22nd chromosomes. In accordance with the position of the centromere, chromosomes are usually divided into 3 groups. Metacentric, the centromere is located in the middle of the chromosome. Submetacentric centromere is placed from the center of the chromosome. Acrocentric. The centromere is located in the distal part of the chromosome. All autosomes, according to the Denver classification, were divided into 7 groups, from A to G. Group A chromosomes from 1st to 3rd, large metacentric chromosomes. Group B chromosomes from 4th and 5th, includes large submetacentric chromosomes. Group C chromosomes from 6th to 12th, medium-sized submetacentric chromosomes. Group D, chromosomes from 13 to 15, large upper-central chromosomes. Group E, chromosomes from 16 to 18, includes short submetacentral chromosomes. Group F, chromosomes from 19 to 20, small metacentral chromosomes. And group G, chromosomes 21st and 22nd, include small acrocentric chromosomes. The sex X chromosome in length and centromeric index, the ratio between the length of the short and long arms of the chromosome, is close to the chromosomes of group C, and the Y chromosome in size and morphology with normal color is close to the chromosomes of group G. In 1970, first, in Paris at the 4th International Congress on Human Genetics, a unified system for identification of human chromosomes was agreed, taking into account the differentiation of chromosomes by length. Each chromosome of human set in differential staining is characterized by a unique combination of dark-colored segments or stripes alternating with unstained areas or light segments. It is this specific combination of segments for a given chromosome that makes it possible to clearly identify it and distinguish it from other chromosomes in the set. Within the short P and long P arms of each chromosome, a number of clearly identifiable regions or regions are distinguished, which are numbered with Arabic numerals starting from the centromere to the telomeric region or terminal end of the chromosome. Each region of the chromosome includes a certain number of segments, the number of which, with a second Arabic numeral, also goes in a direction from the centromere to the telomeric region. Thus, the designation of the chromosomal segment 2q34 means chromosome number 2, long arm, 3 region, and third region, and fourth segment. The centromere itself is designated by a combination of the numbers 1 and 0. Thus, the part of the centromere within the short arm is denoted as T10, and the part including the long arm Q10. The discovery in the middle of the 70s of the fact that prophase and metaphase chromosomes are reaching a larger number of segments than metaphase chromosomes, and therefore increasing the resolving capabilities of cytogenetic research, led to the development of methods for obtaining high-resolution chromosomes and required an addition of the cytogenetic nomenclature. New principles for the analysis of such chromosomes were found. In 1980, an international agreement was reached on this in Paris, which was published in 1981 under the title International System of Cytogenetic Nomenclature of Human Chromosomes, High-Resolution Chromosome Segmentation. So, if a segment within a chromosome is subdivided into separate subsegments, then a dot is put after the segment number after which the subsegment number is indicated. For example, if the original 1p31 segment is subdivided into 3 different subsegments, then they are designated by 1p31 first, second, and third, with the 1p31st one subsegment being proximal and the third subsegment distal to the centromere. Karyotype is a set of features, number, size, and shape of a complete set of chromosomes, inherent in the cells of a given biological species karyotype, a given or any individual karyotype or line clone karyotype of cells. A graphic representation of the karyotype, that is, a set of chromosomes when they are arranged in groups depending on their shape and size, is called an ideogram or karyogram. The appearance of chromosomes changes significantly during the cell cycle. During interphase, chromosomes are localized in the nucleus, as a rule, are despolarized and difficult to observe. Therefore, cells in one of the stages of their division, the metaphase of mitosis, are used to determine the karyotype. For the procedure for determining the karyotype, any population of dividing cells can be used. To determine the human karyotype, every rule of life and the cells are used. The transition of each from their resting stage G0 to proliferation is provoked by the addition of the nitrogen phytohalogen. Bone marrow cells, a primary culture of skin fibroblasts, can also be used to determine their karyotype. To increase the number of cells at the metaphase stage, chromatin or nocodazole are added to the cell culture shortly before fixation, which block the formation of microtubules, thereby preventing the chromatids from diverging to the poles of cell division and the completion of mitosis. After fixation, preparations of metaphase chromosomes are stained and photographed. A so-called systematized karyotype is formed from micrographs. A numbered set of pairs of homologous chromosomes. The images of chromosomes are oriented vertically with short arms upward. The numbering is performed in descending order of size. A pair of sex chromosomes is placed at the end of the set. Historically, the first man-made karyotypes that made it possible to classify according to the morphology of chromosomes were obtained by Minovsky Jensen. Staining, however, further detailing of the structure of chromosomes in the karyotypes became possible with the advent of differential chromosome staining techniques. The most commonly used technique in medical genetics is gene differential chromosome staining. To obtain a correct karyotype, chromosomes are stained with various dyes or mixtures thereof. Due to differences in the binding of the dye to different parts of the chromosomes, the staining occurs unevenly and the characteristic slit structure is formed. A complex of transverse marks English banding. Reflecting the linear heterogeneity of the chromosome and specific for homologous pairs chromosomes and their sections, with the exception of polymorphic regions, various allelic variants of genes are localized. The first chromosome staining method, that allows obtaining such highly detailed images, was developed by the Swedish cytologist Persson, staining. Other dyes are also used. Such techniques are collectively called differential chromosome staining. Q-staining is about staining according to expression with a protein mustard gas examination under fluorescent microscope, most often used for the study of Y chromosomes. Rapid examination of the genetic sex, identification of translocations between the X and Y chromosomes or between the Y chromosome and autosomes. Screening for mosaicism with the participation of Y chromosomes. G-staining, modified Romanovsky gene staining. Their sensitivity is higher than that of Q-staining, therefore it is used as a standard method for cytogenetic analysis. Used when detecting small aberrations on macro chromosomes segmented differently than normal homologous chromosomes. Horse staining. Acridine, orange, and similar dyes are used. At the same time, regions of chromosomes that are insensitive to G staining are stained. It is used to reveal details of homologous G or Q negative regions of sister chromatids or homologous chromosomes. C staining. Used to analyze centromeric regions of chromosomes containing constitutive heterochromatin and variable distal part of the Y chromosome. D staining used to analyze telomeric regions of chromosome. chromosomes. Recently, the so-called spectral karyotyping technique, fluorescence in situ hybridization, or FISH, has been used, which consists in staining chromosomes with a set of fluorescent dyes that bind to specific regions of chromosomes. As a result of such staining, homologous pairs of chromosomes acquire identical spectral characteristics, which not only greatly facilitates the identification of such pairs, but also facilitates the detection of interchromosomal translocations, that is, the movements of regions between chromosomes. The translocated regions have a spectrum that differs from the spectrum of the rest of the chromosome. So, what about the summary of this lesson? It can be indicated that the core carries, that the nucleus carries out two groups of functions: storage of genetic information and its implementation entering protein synthesis. The first group includes the process that conditions the maintenance of hereditary information in the form of an unchanged gene structure. These processes are associated with the presence of so-called repair enzymes that eliminate spontaneous DNA damage, which keeps the structure of DNA molecules practically unchanged in a series of generations of cells or organisms. Further, in the nucleus, reproduction or replication in separation or segregation of DNA molecules take place, which makes it possible for two cells to obtain completely identical volumes of genetic information in a qualitative and quantitative sense. In the nucleus of eukaryotes, the process of change and recombination of the genetic material occur, which is observed during meiosis, the process of crossing-over. The nuclei are involved in the distribution of DNA molecules during cell division. Another group of cellular processes supported by the activity of the nucleus is the creation of its own apparatus for protein synthesis. This is not only the synthesis or transcription of different RNAs and DNA molecules, but also the transcription of all types of transfer RNA and ribosomal RNAs. Maturation, crossing, and splicing of primary transcripts occurs in the nuclei of eukaryotic cells. In the nucleus of eukaryotes, ribosomes subunits are also formed by complexing ribosomal RNA synthesized in the nuclei with ribosomal proteins that are synthesized in the cytoplasm and transferred to the nucleus. The nucleus is not only a receptacle for genetic material, but also the structure where this material is debated and has different functions. So thank you for paying attention and see you next time. Bye bye.